Ever wonder how 6 feet of DNA fits inside a microscopic cell? It is an unbelievable feat of biological engineering. Welcome to the mechanics of DNA packaging. We are breaking down the blueprint of nucleosomes and chromatin. Get ready to see how your body masters the ultimate storage system. Let's look at the math. One human cell holds about 2 meters of DNA. Multiply that by 50 trillion cells in your body. That equals 100 trillion meters of genetic material. That is enough to travel to the sun and back over 300 times. Yet, it all fits neatly inside your microscopic cell nuclei. Mind-blowing. How does nature pack this massive strand without tangling? It uses physics, electrostatic engineering. Your DNA has a negative charge from its phosphate backbone. Spool proteins, called histones, are highly positive. Opposites attract with immense power. This chemical reaction forces the stiff DNA to tightly fold and coil. Meet the fundamental unit of storage, the nucleosome. It starts with an octamer core of 8 proteins, 2 copies each of histones H2A, H2B, H3, and H4. The negative DNA strand wraps around this core exactly 1.65 times. This specific loop holds precisely 146 base pairs of DNA. This perfect architectural repeating unit is unique to eukaryotes. To secure the wrap, nature adds a lock. A single linker protein named H1 steps in. It pulls in 20 more base pairs, making 2 full turns. This complete 166 base pair unit is a chromatosome. Short sections of unwrapped, linker DNA connect them. This primary packaging shortens the original DNA fiber 7-fold. How do scientists know this happens? They proved it using a specialized enzyme called mnase. Mnase targets and cuts the exposed, unprotected linker DNA. By stopping the reaction early, they got fragments of different lengths. Running these on a gel revealed clean mathematical multiples. Definitive proof that chromatin is built from repeating structural units. DNA packaging occurs in a multi-level hierarchy. It starts with 2 nanometer naked helical DNA. It turns into the 11 nanometer beads on a string. Next, those beads coil into a 30 nanometer tightrope. Those ropes form huge 250 to 300 nanometer loops. Finally, they form the 700 nanometer metaphase chromatid arm. Ultimate compression from start to finish. Let's zoom in on that 30 nanometer chromatin fiber. The basic beads on a string layout is still too long for the nucleus. Histone H1 stabilizes the structure, allowing it to coil tighter. Textbook diagrams often show perfectly uniform coils. Real electron micrographs tell a different story. Inside living cells, these 30 nanometer fibers are highly complex and irregular. Extreme compaction solves space problems, but creates a barrier. Your cell needs to read DNA for replication and transcription. To do that, the 2 genetic strands must temporarily separate. But reading enzymes cannot physically access the locked template. It is trapped inside the dense 30 nanometer fiber. How does the cell break through this biological lockdown? The cell overcomes lockdowns with dynamic, reversible mechanisms. First, enzymes use modification tags like acetyl or methyl. These alter the positive grip of histones, loosening the DNA. Second, remodeling complexes physically slide histones away. This exposes target sequences to reading enzymes. Once finished, everything seamlessly returns to its compact state. The absolute peak of compaction occurs during metaphase. DNA reaches a staggering 10,000 to 1 packing ratio. Imagine compressing a football field of rope into a half-inch coil. Why do this? To protect the genetic library during cell division. Loose DNA would instantly tangle, tear, and shear apart. This brilliant folding system ensures safe transit to new cells.